Welcome to our fourth and final video for our Foundation Cell Biology series. In this video, we will take a look at diffusion and osmosis. But before we look at diffusion and osmosis, we need to talk about solutes, solvents, and solutions. Okay? So these are terms you may or may not have heard before, but let's define them using Kool-Aid as an example. What are these three things? Is a liquid that another substance is dissolved in. If you're making Kool-Aid, the water you put in the pitcher, that is the solvent. It's the liquid that everything else is dissolved in. We add to that our solute. Okay? And our solute is what's being dissolved. In the case of Kool-Aid, it is the flavor molecules, the sugar molecules, the preservatives, all of those are what are being dissolved into the water, which is the solvent. When you put these together, you get the solution, okay? And a solution in chemistry is a mixture where one substance, the solute, is dissolved in another liquid, the solvent. Okay, now in biology, that solvent is usually going to be water. It doesn't always have to be, especially in chemistry, but for usually biology, our solvent is water. So, why do we care about this? We're going to get there, but first let's take a look at chemical diffusion. Okay, so chemical diffusion is the random movement of molecules from areas of high concentration to areas of low concentration. Okay, so what that looks like. In this GIF, wait till it restarts. Okay, we start with a dense area of molecules, and they are randomly moving around until, and they're moving to the areas where there weren't any other molecules, until they are evenly spread out. Now, it's important to know that these molecules aren't like you in the lunchroom. They're not saying, ooh, there's an empty spot, let me go grab it. They are just randomly moving around and bumping into each other until they evenly spread out. An example of chemical diffusion, this is why you can smell Chick-fil-A outside of Chick-fil-A. This is why if you put a drop of food coloring into a glass, the whole thing will eventually turn that color, all right? My favorite example is actually from my time in middle school. In 7th grade, there was this kid named Joe. Joe brought a bottle of cologne. We didn't have Axe back then, but it was the kind of strength of Axe. So we'll say that Joe brought some Axe to school, and the bottle of Axe broke in his locker. And by the end of the day, the entire 7th grade wing smelled like Axe, okay? And the way that worked is because the cologne started in a highly concentrated area, namely Joe's locker, and then the molecules randomly spread out until the entire 7th grade wing smelled a little bit less strongly than his locker of Axe, okay? So that's chemical diffusion. What is biological diffusion then? Okay, because in biology, we're not usually just talking about molecules moving randomly. We add a little qualification, right? So biological diffusion is the net movement, which we're going to define as the overall movement. Because remember, the molecules are moving in all directions. It's just that if we sum them up, they're all going to be in one direction. So it's the net movement of solutes from areas of high solute concentration to areas of low solute concentration. So in this GIF, we can see this side starts with more molecules, and then the net or overall movement is towards this other side, which has fewer molecules. However, you'll notice that there are some molecules that are moving the opposite direction, like that molecule went that way, this molecule crossed the membrane this way, okay? So, unfortunately, this isn't the end of our definition. So we got our net movement of solutes from areas of high solute concentration to areas of low solute concentration. What's different here is they are moving across a membrane. Okay, and these molecules will keep moving until they reach equilibrium, and we'll define equilibrium on the next page. But I do want you to remember that molecules are moving randomly and in all directions. And they're moving back and forth across the membrane. They're not just like, ooh, there's low concentration, let's go fill that up. What do we mean when we say that this keeps happening until the molecules reach equilibrium? Equilibrium occurs when there are equal concentrations of a given solute on both sides of the membrane. So in this example, remember, it looks like the molecules are moving this direction, but in reality, some of them are moving this way, some are moving this way, okay? But the net or overall movement is towards the area with no solutes, okay? We go through this stage, okay? And then eventually we reach equilibrium. At equilibrium, the molecules are moving back and forth at equal rates. Okay, and we can say that at equilibrium, the net or overall flow equals 0 because molecules are moving at the same rate in both directions. Okay? But remember, the molecules are still moving. So some properties of simple diffusion. So the simple diffusion we already defined. It's that movement of solutes across a membrane. Okay, they're moving from high to low concentration. And this is passive. It doesn't require any energy from the cell. It just happens. All right, now what kinds of molecules can do simple diffusion? Because it's not all of them, all right? So remember, the cell membrane is semi-permeable, meaning it only lets some things through. So what can a cell membrane let through on its own? It is small molecules, okay, like water, non-polar molecules, like fats. And the reason for this is because it has to get through this non-polar layer in the middle, and polar molecules can't get through that, okay? So most of these small nonpolar molecules are things like water, gases, fats, okay? That's what can get through. What about for the molecules that can't get through on their own? For those, we have facilitated diffusion. So in facilitated diffusion, the molecules still move from high to low concentration. Okay, you can see they move from an area of high concentration to an area of low concentration. But the molecules can't go through on their own, and this is because they are too big or too polar to get through the membrane alone. Examples of these are things like salts and sugars. Those are too big in the case of sugar and too polar in the case of salt to get through. And so normally they would be blocked by the membrane. So how do they actually get across? Well, they get across by moving through protein channels. Remember, there are proteins in the cell membrane. It's not just those phospholipids. Some of those proteins are channels. And lastly, this requires no energy from the cell. Okay, it is passive as well. But then let's take a look at active transport. Now, if you look at our diagram of active transport, there are some very different things about active transport. One is that the solutes move from a low concentration to high concentration. Because of this, active transport requires energy. Okay. The analogy I like to use for this, imagine you were on a slide. If you were at the top of the slide, it would take you no energy to get down the slide. However, if you are one of those kids that started at the bottom of the slide and crawled all the way to the top, that would take you energy, okay? So high to low, no energy. Low to high, does take energy. There are 3 ways to do active transport. One is through protein pumps. That's what is shown in your diagram right here. Okay, there's a protein that literally pumps the molecules across the membrane, and that takes energy, okay? There's 2 other kinds. There is endocytosis and exocytosis. Okay, cyto means cell, endo means into. So endocytosis brings things into the cell, exocytosis puts things out of the cell. And these, they require energy on the cell's part because it requires proteins to make this movement happen. But these are often used for very large materials. Like this is what an amoeba uses to eat another organism, for example. Okay. Now, what does endocytosis and exocytosis look like?
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
All right, we're going to watch that again, actually, and I'm going to point out, I'm going to lower the volume. I'm going to point out some of the cell features that hopefully you saw yesterday during your organelle activity. So here you can see that phospholipid bilayer.
Transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis,
here the membrane is behaving like a fluid. Remember, it's a fluid mosaic.
Which brings the contents into the cell's interior. In the opposite process, called exocytosis.
Okay, so here, this is a vesicle, okay? That small membrane sac used for transporting things. You can't see it, but it's traveling on the cytoskeleton, okay? It was made by the Golgi apparatus.
The sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
All right, so that's endocytosis and exocytosis. Now we are going to move to osmosis. So osmosis is the diffusion of water, that solvent, across a membrane. Okay, this happens when the solutes can't move. So let's say something like, let's say there's a difference in salts on either side of their membrane. Remember, salt is too polar to cross the membrane. So if the salt can't move to equalize the concentrations on both sides, then the water has to move. And so what direction does the water move? So the water moves from areas of high water concentration to areas of low water concentration. All right, so if we look at this diagram right here, okay, sides A and B, B has more water per red solute molecule, okay? And so the water moves from the area with a higher water concentration to the area with a lower water concentration, okay? But usually with osmosis, we don't talk about the water concentration. Instead, we talk about the solute concentration, okay? So if we said that side B has a high water concentration, what you can also see is that it has a low solute concentration. And it moves to the water moves to the side with the high solute concentration. All right, so a good way to remember this is that in osmosis, the water moves toward the solutes. Wherever there are higher solutes, that's where the water is going to go. A lot of times, as we're talking about osmosis, we talk about tonicity, all right? So tonicity is the solute concentration relative to the other side of the membrane. All right, what do I mean by this? So let's say you weren't given both sides of this. Okay, let's say you just had one container right here. Okay, you couldn't say that this has a high or low concentration because you have nothing to compare it to. It's only when we compare this side to this side that we can say that one side has a higher concentration. And one side has a lower concentration. It only works if you've got a comparison. We're going to look at an example, and the example is going to use red blood cells. So for our example, just as a reference point, this is what your red blood cells look like. They kind of look like little flattened donuts where all the jelly has been squished out of the middle. Okay, so that's what they normally look like in your blood. We have 3 words for tonicity that we will typically use, okay, and we usually have it refer to the solution. So this jar right here, okay, if you'll notice, there is a lower concentration of solutes outside than inside the cell. Okay, so we call this a hypotonic solution. Okay. Like we said, the hypotonic solution has a low solute concentration outside the cell and a high solute concentration inside. The best way to remember this is that hypo and low go together. Okay. If you'll notice, if we count the number of arrows, more of the arrows and more of the water is moving into the cell. So the net water movement is into the cell. And because of that, the cell swells up. All right. This does not look a little flat, like a little flat donut anymore. This would look more like a kickball from school. Okay. It is big, it is round, it is inflated. On the opposite end of the spectrum, we have a hypertonic solution. Right. And if you'll notice in this hypertonic solution, the concentration of solutes is higher outside and lower inside. You'll notice that our net water movement is out of the cell. And as a result, the cell shrivels or shrinks. You actually experience this a little bit when you eat potato chips. The salt in the potato chips draws the water out of your mouth and tongue cells, so you get a little bit dehydrated in your mouth. So then lastly, we have this other solution, okay? This solution is called isotonic, okay? And in an isotonic solution, there is an equal concentration of solutes in and out of the cell. In other words, we could say that in an isotonic solution, the cell is at equilibrium. And remember that when something is at equilibrium, the net water movement is 0, and the cell stays the same. It does not grow, and it does not shrink. Now remember, this doesn't mean that water molecules aren't moving at all. They're just moving at equal rates into and out of the cell. What does this look like? Here's a brief animation. You can see in our hypotonic solution, the water is moving into the cell, and the cell swells up. In our hypertonic solution, the water is leaving the cell, okay, and the cell shrinks. And in isotonic, they're moving in and out at equal rates. Remember that in all of these, the water is moving toward the solutes. So if the cell is inflating, that means there were more solutes inside it. And if the cell is shrinking, it means there were more solutes outside it, and the water is moving out to join the solutes. Last but not least, let's compare passive transport and active transport. You'll remember passive transport is the kind that requires no energy, whereas active transport needs energy. And that energy is in the form of ATP. Okay, remember that passive transport typically moves from low to high concentrations, whereas active transport moves from high to low or is for very large materials. Like if one cell is trying to eat another. Okay. For both of them, we've got 3 kinds. Our 3 kinds of passive transport are simple diffusion, facilitated diffusion, and osmosis. Whereas for active transport, our 3 kinds are protein pumps, endocytosis, and exocytosis. All right, we have made it to the end. So for your knowledge check, let me be upfront. It took me until I had to teach about osmosis in order to understand it. So know that this is a confusing concept. If you're not understanding it, that is totally fine. Please ask questions. But this is our knowledge check. This is what you should be able to answer, okay? If you pour some sugar into a cup of water and mix until the sugar dissolves, which one's a solute, which is a solvent, and which is a solution? What direction do solute molecules move during diffusion? What is their net or overall movement? What kinds of molecules can pass through the membrane in simple diffusion? How is facilitated diffusion different? What are the types of active transport? How is osmosis similar to diffusion, and how is it different? You put a cell in pure water. What kind of solution is the water, and what will happen to the cell, and why? And then lastly, why does active transport need energy, but passive transport doesn't? If you feel solid on these, great, you can move on. If not, go back and watch the video, or come prepared with questions for your live learning session. That is it for osmosis and diffusion. Thank you so much for attending these Foundation Cell Biology lectures. We look forward to seeing you in the live learning sessions. And again, please contact us with questions.